Hello everyone. I will be presenting the development of a human nose on a chip model as a physiologically relevant platform for in vitro nasal drug testing. Intranasal administration attracts increasing attention for delivery of drugs and vaccines due to the numerous advantages offered by the nasal mucosa, such as the highly vascularized large surface area for drug absorption, avoidance of first-pass hepatic metabolism, and the direct access to the brain tissue through the olfactory region in the nasal cavity. Hence, nasal drug delivery can be used for localized therapies, drug delivery to the systemic circulation, and the central nervous system. However, this interesting route of drug delivery is limited by the mucociliary clearance that clears the mucus layer every 15 to 20 minutes, resulting in the short residential time of inhaled drug particles on the nasal mucosa. Also, the nasal epithelial barrier function limits the transport of drugs across the epithelium. All these factors are considered for development of novel nasal drug formulations for efficient nasal drug delivery. The efficacy of novel nasal formulations is tested during preclinical studies by in vitro cell culture models where transval or snapple inserts are mostly used to culture nasal epithelial cells under air-liquid interface condition. Also, the donor-acceptor structure of these cell culture platforms allows for assessing the transport of drug across the nasal epithelium model. However, these platforms only provide a static condition that is not relevant to the dynamic microenvironment of human nasal mucosa, and also they require sampling and costly, time-consuming analytical procedures using large volumes of reagents. Hence, there is a need for development of an alternative platform for testing nasal drugs in vitro that is not only physiologically relevant, but also enables time and cost-effective drug testing assays. One solution is the application of microfluidic technology or organ-on-chips that are miniaturized devices that emulate the dynamic microenvironment of human organ tissues, including the fluid flow, mechanical forces, and stresses. These features can enhance the relevance of in vitro drug tests on these platforms and as a result improve the predictability of clinical outcomes. In this research, we have developed a human nose on a chip by fabricating a microfluidic device that allows for air-liquid interface cell culture, emulation of pulsatile systemic circulation, as well as the realistic flow of nasal drug particles. Moreover, the sensors integrated in the nose on a chip allow for monitoring formation of tight junctions by measuring transepithelial electrical resistance. Also, the carbon nanofibers modified carbon electrode in the acceptor microchannel allows for quantifying the permeated drug to the acceptor microchannel electrochemically in real time. By the culture of human nasal epithelial cells in the nose on a chip, this device mimics the characteristics of the human nasal mucosa in terms of the mucus production, epithelial barrier function, and transepithelial electrical resistance. In addition to that, the nose on a chip allows for simulation of nasal drug particles delivery to the cells under dynamic conditions. To do so, the inlet of the donor microchannel was connected to a nebulizer and the outlet was connected to a vacuum pump to apply a flow rate of 0.5 liters per minute. The deposition of sodium fluorescein dye aerosol to the nose on a chip showed that the particles delivered to the nose on a chip have a range of diameter between 1 to 30 micrometers with median of 7 micrometers. The integrity of the cell layer was maintained under this dynamic condition as the transepithelial electrical resistance did not show any significant difference before and after aerosol deposition in the nose on a chip. Comparison of the nose on a chip with human nasal cavity shows that the flow-induced shear stress in the nose on a chip lies within the range of shear stress reported for human nasal cavity. In addition, the size of particles delivered to the nose on a chip matches the size of particles delivered to the olfactory region for nose-to-brain drug delivery. In this study, we have used ibuprofen as a model drug because of its potential effect on management of Alzheimer's disease. The pathogenesis of Alzheimer's disease is associated with secretion of inflammatory cytokines in the brain tissue due to the deposition of beta-amyloid plaques on the neuron cells. Hence, the anti-inflammatory drugs, including ibuprofen, can postpone the onset of Alzheimer's disease. Animal studies have shown that orally administered ibuprofen had a very low permeation to brain tissue. Thereby, there is an increasing interest in administration of ibuprofen intranasally for efficient brain targeting. To evaluate the effect of dynamic flow condition on the nasal drug testing outcome, drug transport study was performed under three different experimental conditions by the nose on a chip. First, the drug transport was performed under static condition similar to snap flow inserts, and there was no flow in either donor or acceptor microchannel. In the second experiment, the systemic circulation was simulated by applying a pulsatile flow through the acceptor microchannel. In the third condition, in addition to the pulsatile flow in the acceptor microchannel, a flow of aerosolized drug particles was also simulated in the donor microchannel. The transported ibuprofen was quantified by the carbon nanofibers modified carbon electrode in the nose on a chip, and the profile of the cumulative transported drug mass is plotted. Comparison of the first two experiments shows that the application of a pulsatile flow through the acceptor microchannel significantly increased the rate of drug transport. This could be related to the effect of the flow condition on the mass transfer coefficient in the bulk liquid. In the third experiment, the application of drug aerosol flow in the donor microchannel in addition to the pulsatile flow in the acceptor microchannel resulted in a significantly lower rate of drug transport. This can be related to the lower mass of drug delivered to the channel by the aerosol particles, the heterogeneity of the particles deposition on the cell layer, and the wetting and diffusion of the particles into the mucus layer before getting transported. This is also in agreement with a 2-hour lag that was observed before the gradual increase in the drug mass. The drug quantification by the carbon nanofibers modified carbon electrode was validated versus HPLC for all the three experiments, showing the reliability of the measurements by the nose on a chip. In conclusion, a nose-on-a-chip device is successfully developed for in vitro nasal drug tests that offers multiple benefits such as enhancing the relevance of nasal drug tests by including the effects of dynamic flow conditions and nasal drug particles delivery. Moreover, it offers a validated in situ drug quantification method that is time efficient and also adaptable for quantification of other drug compounds. I would like to appreciate the leaders and members of the respiratory technology team at the Woolcock Institute of Medical Research, as well as the School of Engineering at Macquarie University for their support and generosity in sharing their knowledge. Thank you for your time and attention.
Thank you very much. That was a very interesting talk, and I think certainly is stimulating quite a lot of questions this end. So I'd like to start off, if I may, Hanieh. Can you comment a little bit on, you mentioned that ibuprofen has been demonstrated to have some potential efficacy in Alzheimer's. Can you talk to us a little bit more around the general acceptability of ibuprofen for delivery in the respiratory tract? Normally, quite often, non-steroidals are generally best avoided, particularly in patients who may have respiratory compromise.
So are you referring to administration of ibuprofen intranasally or its effect on Alzheimer's disease treatment?
Intranasally, and the potential impact on it being delivered to the respiratory tract as a consequence.
Ibuprofen has been, there is actually quite much interest in studies of delivering anti-inflammatory drug, including ibuprofen intranasally. And one of the reasons is, as reported in the literature and the studies that are reported, the main interest is for nose-to-brain drug delivery, as I mentioned in the presentation. But in terms of having in vivo studies, as I mentioned, like orally administration have been studied. However, I think it is being studied, but I'm not sure if there is currently a product already tested in vivo or not. But yeah, as I mentioned.
So super, thank you, Hanieh, for that. I'm going to go to Ben and Alex in the other studio, and because I think they may have a question or two. Ben, Alex.
Yeah, I've got a question. I'm intrigued at the innovation you've got where you're measuring transepithelial electrical resistance. And can you explain how does that work from an air interface to a liquid? So how do you manage to measure the resistance in that configuration?
Yeah, actually, true. That's a valid question. So normally for a measurement of electrical resistance, we need the liquid to be at both sides of the membrane. So the cells are cultured under air-liquid interface throughout the cell culture period. But at the time of the measurement of the electrical resistance, we will inject some liquid in the top microchannel as well, and then do the measurement, and then that liquid will be emptied from the top microchannel. So the cells are cultured on the air-liquid interface, but at the time of the measurement, we need to inject some liquid on top as well.
Okay, and does that leave the air interface sort of mucosa in a suitable condition? Does it leave the mucus lining intact and those sorts of things that will be important for your studies?
So what we have been observed during our experiments was that, like, it depends on the flow rate of the liquid over the cells. So if the liquid is gonna be, if the flow rate of the liquid is too high over the cells, we could see some like negative effects. So that's why we had to consider this throughout our studies, to flow this liquid at a very low flow rate so that we do not make any harm to the cells. So yes, what we had we had observed during experiments was that we needed to optimize this flow rate, but I can say that during our measurements we had a very low flow rate to inject the liquid on top of the cells. And once the cells are formed, when the tight junctions are formed and the mucus layer is formed, the possibility of harming the cell layer is too low. So also it is also approved by measuring our TER throughout time. So we could see a significant increase in TER. That means that we are not hurting the cells every time by our measurement. So that also is one positive sign for us when doing
Hanieh, thanks very much, Ben. Thank you for those questions. So Hanieh, just a final question from me and one from Michelle. I think, Michelle, you're going to ask about next steps. But what I just wanted to ask is, so we understand the epidemiological observations in people who have rheumatoid arthritis. They've been given ibuprofen, and there's a decreased risk of Alzheimer's compared to those who are not on ibuprofen. What's the mechanism of action? What has been purported as the mechanism of action of ibuprofen with respect to Alzheimer's?
I believe it's the anti-inflammatory, actually the main function of ibuprofen, which is effect. So the deposition of beta amyloid peptide plaques on the neural cells, which causes the secretion of inflammatory cytokines. That's where the anti-inflammatory effect of ibuprofen can actually postpone the onset of Alzheimer's disease. It is still being studied how it can treat the Alzheimer's disease, but what is reported in the literature is its effect in postponing the onset of Alzheimer's disease development. So that's...
So Michelle.
Yes, finally, thank you very much, Hanieh. Last question really is, what next? I mean, where would you see this technology going towards moving towards clinical application. Can you tell us how you would see this being progressed?
Of course, thank you for this question. So as mentioned, like the main point for this study is to enhance the relevance of nasal drug test in vitro. So our first step was to simulate the nose on a chip and also improve the drug test. But we are also going for simulating diseases such as nasal wounds or infections, and also testing the efficacy of drug in treatment of diseases. So this will be our next step, which I think will prove like how we can test the efficacy of drugs on chip.
Thank you very much, Hanieh.